All right, so it is Monday. It is week 3 already, and here we go. It's winter 2021. Now this is not in your slides, but I have some 7th quarter students studying for the boards, part 1. There's a board book that Palmer puts out, and it's got all sorts of errors in it. So I've been getting all sorts of questions. So I just sent this to this group of students who are studying this stuff because I can't go over everything. But I'll teach you the basics so you understand when you have to do some self-study. You'll be able to understand what you're reading at least. This is Langman. I talk about a lot. It's medical endocrinology or embryology. I keep saying it, endocrinology. It's the 14th edition, and it's not that terribly. I mean, you can rent the thing for like $19. Yes. Oh, you can't. The Oh, okay, yeah, it's not working then. I'm sorry about that. I'll put it on YouTube. You just have to maybe go eat breakfast. I'm not going to take any attendance then. If you want to, yeah, go eat breakfast because it's not, whatever reason, it's not working. I restarted it 3 times. So sorry about that, guys. So you just watch this on YouTube. Go have breakfast. I'll be done in about an hour with this. Or you can, I mean, listen, I guess. You can listen and follow along with the slides is another way to do it. But sorry about that. Let me get through this though. But this is Langman's, and this is the one I would go with because I was on this, the National Board Test Writing Committee. I still am actually. And this is the one that seems to be the favorite of everybody. So anyway, that's not on there. But here we go. This is, I guess this would be your slide number 2. Meet the ovulated oocyte. So we kind of know this one already. And yep, there it is. That was released. If you watch last week's lecture, you know all about this oocyte, how it was created. And it's haploid. It has only 23 chromosomes. It has the half number of chromosomes. And normally, after it's ovulated, if it comes out of the ovary, it's swept out by the fimbriae and kind of swept into the infundibulum down the fallopian tube. And remember that it gets stuck in metaphase 2 of meiosis, so that's an important thing, a nice, easy thing for people to get tested on. Stuck in metaphase 2, and it won't start. It'll never start again. So your secondary oocytes will not ever come to full maturation unless a sperm penetrates them, and that will restart metaphase 2, as we'll see. All right, so that's an interesting kind of big picture concept. So now the sperm, it's easier said than done to fertilize an egg. There's 3 barriers that you have to get through if you're a sperm and you're trying to get in a secondary oocyte, which is usually, remember, in the ampulla of the fallopian tube is where the meeting usually occurs. So you have to get through the corona radiata, pretty easy. The zona pellucida, not so easy. And the vitalin or vitelline membrane, that's a fancy AKA for just a run-of-the-mill plasma membrane or cell membrane. That's the oocyte's plasma membrane. They call it the vitalin membrane, but it's the same thing. So that's the job of the sperm to get through those things. Here's a picture of the corona radiata. It's got 1 to 3 layers thick. That's not shown here, maybe 2 layers in this picture, but it's a little thicker than that. And there's the zona pellucida, this little yellow layer right here, and that little red layer, that is actually the plasma membrane, or the vitalin membrane, or the cell membrane, whatever you want to call it. Got to know those AKAs though, because you never know what the board question will ask. So how does the sperm get through these barriers? What is its weapons? There's 2 processes. It has the capacitation phenomenon that has to occur, and that matures the sperm, and we'll see how that works. And then the acrosome reaction, because the acrosome releases some very important juices, really enzymes that help it get through. So capacitation, let's talk about first. This is considered the final step in sperm maturation, and it occurs when the swimming sperm's plasma membrane bumps into the lumen of the fallopian tube. Remember, the sperm is swimming, and it goes through the cervix into the uterus, the body of the uterus, and then it goes into those little tubes, both little tubes. One will have an egg, one will not, because only one. You ovulate, your ovaries take turns spitting out an egg or ovulating an egg, a secondary oocyte, each month. So as it's swimming down the tube, that lumen is called the oviduct. That oviduct, the cells of the oviduct release some factor that they can't really figure out what it is yet, so it's unnamed yet. And that factor is what soaks into the sperm and starts the process of capacitation, of final maturation. So what does that mean? What exactly happens when that factor leaks in? Well, there's two phases of it. The first thing that happens is the sperm's plasma membrane, or cell membrane, or whatever you want to call it. It unloads cholesterol molecules. It dumps cholesterol. So cholesterol just gets thrown into the lumen, kind of like dropping off bombs, I guess. If you're a B-52 bomber back in the day, you're unloading cholesterol. Think of that as unloading. And that, because normally cholesterol prevents fertilization from occurring. You can't bind to the Zona Pellucida with cholesterol. So that's gone. And then right after that, there's glycoproteins, which we won't even worry about. We'll just call them glycoproteins. Those get unloaded as well. So now the sperm is mature and ready to bind with capacitation. Another thing that happens once it dumps off cholesterol and glycoproteins, go back to the B-52 bomber, it's much lighter, and therefore it's more nimble, and it gets almost hyperactive. It can move really fast, and you can tell that capacitation has occurred when the little tail starts beating really fast, and it's almost like a hyperactive phase. And the tail waggling faster gives it more forward momentum. It's like a boat now, or the propellers of the B-52 turning faster. It can go faster because it's lighter. It's not really because it's lighter, but I mean, that's the analogy. And therefore it can drive harder into when it finds the egg, it can start to push really hard through the Zona Pellucida and Corona Radiata, and then through the Zona Pellucida. So the Corona Radiata we have to take on first. That's the outermost barrier. Remember last week's lecture, this came from embryological development. It came when the secondary oocyte was expelled during ovulation. It took this Corona Radiata and the Zona Pellucida with it. It used to be called the cumulus operus layer, so it's still there with it. And it has space between the cells, so you could think of the space between the Corona Radiata cells as an intercellular matrix, or an interstitium basically. And it's really rich in hyaluronic acid, which we are going to study in spinal anatomy. So what? And here's just a back to the future view. Here there's the cumulus operus, which is now the Zona Pellucida. Here's our little egg that we talk about, which is really a secondary oocyte. And that's going to be expelled out of the ovary and sucked up into the fallopian tube. This thing is, remember from last week, this thing ruptures and spits this thing right out. The fimbriae grab that. But when it spits it out, it takes the Corona Radiata, and more importantly the Zona Pellucida, which is in brown here. It takes that with it. Alright, so the Corona Radiata turns out to be not much challenge for the sperm. The acrosome, which is now light and ready for action, because we got rid of those things, the cholesterol and glycoproteins, it releases a powerful enzyme called hyaluronidase. And whenever you see ASE, that's an enzyme and it usually cuts. So that's exactly what it does. It doesn't cut through the cells. Sorry about my voice. I don't know what's going on with my voice lately. It cuts through the hyaluronic acid. It doesn't cut through the cell though. It goes through the sides of the cell. And yep, that's what happens. So it gets through, sneaks through the cracks, and you can see the picture of the spermatozoa, or the little sperms pushing right through. They don't dare to go through the cell itself. It just goes through the cracks. So now it bumps into the next opponent, and that's the Zona Pellucida, and that one is much tougher to get through. And you can't even get through that one unless you're able to bind to it. We've talked about this already. These Zona Pellucida glycoproteins, ZP3 proteins, ZP1, 2, and 3, they're also known as Zona Pellucida sperm binding proteins. But ZP3 is the main one we're going to worry about. And that's the one that, remember, the SED1 receptor on the sperm head will bind to the Zona Pellucida 3. And that's how they dock up. So some enzymes can be released. So that's an important one to know. So Zona Pellucida is the main receptor that the incoming sperm will attach to. Getting through the Zona Pellucida. So now that what we have to attach here, so here's the sperm has a SED1 protein, and it binds to the ZP3 protein. We said that. And again, if you have a mutation in any of your genes that make these things, you won't be able to get pregnant. So you can't have SED1 mutations or Zona Pellucida 3 mutations, or it's really, really tough to get pregnant. But the binding triggers something called the acrosome reaction. What is that? I think we've seen this before. There is SED1 sticking out, and there is, the blue ones are ZP3. So SED1 is this long little kind of snake tongue, and it's binding to the blue ZP3. There's also a GAL-T that can also bind to the ZP3. It's not as effective, but it's kind of a backup. So there is two ways to actually bind to ZP3. Okay, so what's this acrosome reaction? So we've bound together, and it's triggered a reaction. And there's a whole bunch of enzymes that are released and soak into the Zona Pellucida after this binding occurs. I cut out most of them, but I do want you to know one of them. Kind of the ringleader, the king of these cutting enzymes, is called Acrosome. Acrosome. And there's more. That's also a board book. It's the most important of them. It's a serine protease, so it cuts protein at the serine amino acid. And yep. So it cuts the acrosome between the cells of the Zona Pellucida. Again, we're not going to go through a Zona Pellucida cell, but we just kind of push our way through the ZP proteins, and the sperm can now start to get in. So it's similar to going through the corona radiata, where you push between the cells and you don't rip right through them. But you need a functioning acrosome gene in your sperm, or that's another way that a guy might not be infertile if that gene isn't working. So here's the Zona Pellucida in this picture. The sperm head is pushing through, and now we're in this space between the plasma membrane of the oocyte, or the vitelline membrane, or what's the other AKA, or the, all those AKAs. But there's a space here where the little sperm head kind of sticking into this space. This is called the perivitelline space, because it's in between the plasma membrane and the Zona Pellucida. All right. Now this is important. There's a lot of stars here for this class. It's important because now fertilization has begun, And so if the board question is when does fertilization begin, it's when the zone gets through the Zona Pellucida, not when the zone gets through and inside the egg. So it's already begun once you get through the Zona Pellucida, so make sure you know that one. And maybe it'll say, you know, it's in the perivitalin space or vitelline space, however you want to say that, tomatoes. But now the clock starts ticking. Fertilization is a process. It doesn't just happen all at once. But the process has now begun. It takes about 24 hours for the process of fertilization to occur. Right, so the next thing that happens is the plasma membrane of the oocyte, still a secondary oocyte, it has these little pegs sticking up here. Those are called microvilli. You'll look at those again if you haven't already. Dr. Doe will talk about those in enterocytes in the small and large intestine. Also has these microvilli, and they increase the surface area. And they grab the sperm and kind of wrap around it. And the plasma membrane of the sperm and the plasma membrane of the oocyte dissolve. And you'll have to look on YouTube. See, by the way, I'm on slide 20. I guess it would be your 21. Or maybe your 19. I shouldn't have put that. If I knew this wouldn't have worked, I wouldn't have put that extra slide in there. But the membranes dissolve right here. And now the sperm can slip inside the oocyte. So that's kind of the deal there. There are unfortunately some receptors that are not shown in this picture. So probably less of a high yield board question. That's where I put the little kind of extra credit guy here, the little nerd. But there are some receptors here. So on the sperm's plasma membrane, there's the azumo atoms 1, 2, and 3 are the receptors that bind to the oocytes receptors. And yeah, they're stimulated, they're put in place when the acrosome reaction occurs. They're kind of laid down, and they're not on run-of-the-mill sperm. It has to go through the acrosome reaction for them to occur. And then on the oocyte's plasma membrane, we have CD9, CD8, Juno, and a bunch of integrin proteins. We won't really worry about that. But CD8 and 9 and Juno meet up with atoms 1, 2, and 3, and azumo. Not fun stuff to memorize, and I know, and I don't know what to do about it. It just kind of is what it is. So now we have part of the sperm in it's gotten into the oocyte. So that's kind of neat. But after binding, the oocyte's plasma membrane is basically swallowed. How does it get in there? It's via endocytosis-like process. But it's really weird. The sperm's plasma membrane doesn't go inside the oocyte. So that's another good easy board question. It's kind of the guts of the sperm that go into the oocyte. The plasma membrane doesn't. It's stuck on the outside. And the mitochondria as well doesn't get in. So it's kind of part of the sperm gets in there. The most important part, which is the nucleus, that's where the genetic material is. That definitely gets in. But on this picture, I guess would be on slide 22, because I have one more slide than you. On slide 22, you can see how this goes. So here's the plasma membrane of the sperm in white here. And there's the kind of the ghost of the sperm getting in. There's the nucleus. That's what we want to get in. But it still has a sort of a tail, but it doesn't really function very good. It's just kind of floating around. Pretty soon we'll have microtubules will grab that sperm head and from the oocyte's nucleus, and it pulls these two together. The sperm little tail doesn't work anymore. So, fall down one of my famous rabbit holes. We should talk about polyspermy at this time. So, how come only one sperm? We have thousands and thousands of sperm trying to get in. How come only one sperm usually gets in? And that's how do we prevent polyspermy? And that's what we'll talk about right now. There's actually two mechanisms that prevent two sperms from getting in the egg. And there's fast block polyspermy and slow block polyspermy. Fast block is the first one we'll talk about, fast block polyspermy. So when the sperm gets through the plasma membrane of the oocyte, something physiologically weird happens to it. It depolarizes like crazy. It goes from 70 millivolt, negative 70, which is a normal membrane voltage, to plus 10 millivolts. So that's really, really strange. So you get a massive depolarization. And that, in and of itself, discourages other spermatozoa from hooking to the plasma membrane of the oocyte. They can't; those receptors can't bind when the plasma or when the membrane is depolarized. Man, my voice is bad this morning. Sorry about that. Let me hit the mic here and take some water, or see if I could. Hopefully you didn't hear me clearing my throat there. I think I figured out how to turn off this mic. Anyway, yeah, that's what happens. So that's fast block polyspermy. So undergoes a massive depolarization. So there's another mechanism called slow block polyspermy. And this involves the release of calcium from the endoplasmic reticulum. So once the sperm gets into the cytoplasm through mechanisms we won't talk about, well, I guess we will talk a little bit about it, but we're not going to go crazy into this. It releases the sperm, kind of the remnants of the sperm, releases an enzyme called phospholipase C zeta. And that binds to the endoplasmic reticulum and causes it, of the oocyte, and causes it to dump tons of calcium out of the endoplasmic reticulum. When you get in physiology, you'll talk about that, especially in muscles and smooth muscles. The dumping of calcium out of the endoplasmic reticulum will cause the muscle to contract. And so, but in this case, it doesn't cause anything to contract. But what it does do, the wave of calcium that kind of shoots out of this structure on slide 25, this endoplasmic reticulum here, this wave of calcium hits the nucleus of the oocyte, and it stimulates it to start meiosis once again. Remember, it's stuck, the secondary oocyte is still stuck in metaphase 2. And so finally, we get to finish meiosis. And in addition to that, it causes these cortical granules that are floating around in the cytosol of the oocyte, causes them to bind to the plasma membrane and release their payload, release their bombs, if you want to stay with that analogy, into that perivitelline space. And we should talk about these cortical granules a little bit. So they're floating around in the cytosol of the oocyte, we said. When the wave of calcium hits them, it stimulates them to bind to the cell membrane of the oocyte and dump their payload, dump their bombs into the perivitelline space. And their bombs are called, we'll just call them polysaccharides, and a bunch of proteolytic enzymes, because you got a bunch of stuff to remember, with no sense of memorizing all these. But it does dump a bunch of polysaccharides and enzymes into the perivitelline space. And this is after one sperm has got in. So what do these things do? Why does it do that? Polysaccharides will soak into the zona pellucida and cause it to get very stiff. And in fact, even the hole where the sperm penetrated will be closed by this reaction. and therefore if it's so stiff that a second sperm will have great difficulty getting through the zona pellucida because of these polysaccharides. Sometimes this in and of itself, this reaction with the polysaccharides, sometimes it's called the zona reaction. And zona reaction is part of slow block polyspermy. And that's the one that the calcium wave stimulates the release of these cortical granules. So what else? So we just said what happens to the polysaccharides. They stiffen the zona pellucida. What about these proteolytic enzymes? What do? So they actually flat out destroy ZP3 receptors on the zona pellucida. And now the sperm can't bind. So that's kind of the second mechanism there. So both of these mechanisms, which collectively are called slow block polyspermy, both of them hinder the ability for a second sperm to get inside the egg. In other words, they inhibit polyspermy. All right, so after penetration, what we said that the wave of calcium stimulates meiosis to get going. So that's awesome. So once meiosis 2 is complete, and this is going on in the nucleus of the oocyte, the secondary oocyte, so it just runs through the rest of meiosis like it does. And it creates, out of that nucleus, we get another polar body, because the other one was degenerated away, but this one will degenerate away. But our secondary oocyte with this new nucleus gets a new name, and it's called the definitive oocyte, or ootid. So that's an important concept. That secondary oocyte, which was ovulated about the 10th day of the month on the female menstrual cycle, that's a secondary oocyte that's ovulated. It'll never become mature unless a sperm gets inside of it. If a sperm gets inside of it, that secondary oocyte matures into the final product, which is a definitive oocyte, or you can call it the ootid. I have a feeling it's going to be called the ootid because that's a board book Moore's. Okay, and that's just a picture of anaphase 2 going on, and we're getting closer to forming the mature ootid. And so this you should know this. I mean, I don't know if you already do know this, but this is kind of where we've been, where we've already talked about the primary germ cells morphing into the oogonia. This is happening before you're even born, and then we get crossover occurs, and we start go through meiosis 1, but they originally get stuck in prophase 1, stuck right there, so they're primary oocytes. And then we said that these can stay with you for 40 years stuck as a primary oocyte in the ovary. And then we said after about the age of 12 or so, every month a bunch of them, maybe 50 of them, wake up, and that starts the great race. And then they go through meiosis 1. As they go through the great race, the winners of the great race, the big three, will actually make it all the way to metaphase 2 and get stuck again. And then ovulation occurs, and if they get fertilized, only then do they continue through anaphase, telophase, and cytokinesis. And this is where we are right here in our story. We've created this mature oocyte, or ootid, aka definitive oocyte. So this would only happen inside when the sperm is inside. Maybe the sperm's nucleus is in here as well somewhere. And this whole thing, what they don't show in these drawings, this whole thing is still wrapped by the zona pellucida and the corona radiata, and the sperm, but the sperm nucleus is definitely in here. Oogenesis is very confusing because you don't know the whole story, and nobody seems to want to teach that right or take the time to actually figure it out. I haven't found a good YouTube video really that explains that. So anyway, there's where we are. And that's just kind of a little review slide. All right, so now we have a mature ootid, or a definitive oocyte, if you will. It's a weird cell because it's a 2 nuclei cell at this point, and we're still in the process of fertilization. It's a 2 nuclei cell, but each nuclei is 1 and 23 chromosomes. So you can see here, this is the male. This is what the sperm's tail was pretty much degenerated, but maybe it has a little piece of tail still. And there's the nucleus, which has already went through meiosis and is mature now. And you can see all the genetic material. I'm on slide 32. And you can see where they're touching right here. And this is going to soon dissolve, and all this genetic material, 23 chromosomes here, 23 chromosomes there, they're going to all mix together. And we're going to create a little human called a zygote at that point. That's where we're going. So coming together, everything I just said. 2 nuclei are pulled together. By the way, when the nucleus has went through meiosis 2, it's called a pronuclei. So the 2 pronuclei are pulled together by microtubules. Let's go back to that picture. You can't really see the microtubules between the 2 nuclei, but they're there. They've pulled them together. And the goal is to merge the genetic material, right? Each one of those nuclei has 23 chromosomes, and we want to make them 46 chromosomes. So we need to move those together. Let's see. So switching to mitosis. So this is interesting. So as these, let's go back to that picture on slide 32. So after metaphase 2 has accomplished, these nuclei actually, as they're coming together, they start mitosis by going through an interphase. Because remember, in interphase, you can't see the chromosomes. These are definitely, these chromosomes in this picture are thick, and we can see them. So these are in metaphase. They're lining up here. Yep, these are metaphase chromosomes. These have already went through interphase and replicated. So that's kind of weird. We have we have 2 nuclei going through interphase at the same time. And so that's just kind of a weird type thing. So they've finished interphase, and I could have added here, they're actually in metaphase as they mix together. On slide 34 now. So yeah, so that's the story there. So coming together, and so as I said, the fusion, they're going to mix their genetic material and become 46 chromosomes. We have already said that's the famous Beetle album there in 35, slide 35. Yeah, our clock is ticking. Fertilization is almost done. And here's just kind of backing up picture on 36 here. There's the sperm. You can see its non-functional tail there. You don't see the microtubules, but there would be thin tubules pulling these guys together. And yep, so interphase is past. I mean, we're already in prophase of mitosis prophase at this point. Okay, coming together again. I could probably cut some of these slides out because I'm really saying the same thing. But yeah, the contact where great picture, right? You can see this sperm. You can't see its tail, but its tail will be degenerated. The sperm and the egg nuclei are coming together. Where's this happening? This is the Zona Pellucida, is right here, and this is the corona radiata, kind of in this weird. We've got some volume averaging on this picture. You can't see the individual cells, but this is still occurring in that oocyte that was spit out during ovulation. So this is the nuclei are the ones that are coming together. All right. So this membrane's gonna be gone, dissolved, and the genetic material are going to be mixed. On slide 38 now, you can see the genetic material mixing. Once the genetic material have mixed and that membrane between the two nuclei is dissolved, then we finally get our little creature. This is where most scientists think is the beginning of life, and this is called the zygote. Okay, so yeah, the two pronuclei going inside this oocyte, and officially this is now a ootid, right? Or the definitive oocyte, because it's mature, because its nucleus went through finally finished meiosis. So yeah, so we have a zygote here. So remember, it's the ootid that morphs into the zygote, right? It's not the secondary oocyte or the primary oocyte. There's a question right there. I know that's on the test. Forgot to put a star there. So it's the ootid, the mature egg that has a sperm inside of it, that morphs into the zygote. So once the zygote is created, the process of fertilization is complete. Slide 39. Okay, so again, once the zygote is complete, process of fertilization is complete. All right, so now we've got a real cell. We've got a cell that is diploid. We've combined two haploid cells to make a diploid cell called the zygote. It's got 46 chromosomes in that cell specifically, or you could say it's got 23 pairs of chromosomes. You have to be careful when you're reading these types of questions. All right, some general terminology. So once the zygote is made, we have entered the first stage of human development, which is called embryogenesis. That'll run for about 8 weeks. We talked about that in week 1 already. Some authors call this organogenesis, so watch out for that. Morse calls it organogenesis. And Morse is a chiropractic board book. 8 through birth is called the fetal period, which we're not even going to come close to getting into. What is fertilization? What have we accomplished? Well, we stimulated the ovulated secondary oocyte to mature by sticking a sperm inside of it, and it became an ootid, or a definitive oocyte. So we've matured the oocyte to its fullest. We've also mixed the sperm and the egg's genetic material to create a real human cell, which has the correct number of chromosomes. It has 46 chromosomes, or 23 pairs of chromosomes, AKAs for each other. Its diploid number is 2n, and we've created the first little human called a zygote. And we've mixed the chromosomes up. Mom and dad's chromosomes have mixed together. Some of them will be silent, some of them will be expressed. Remember, there's 2 pairs, one for mom and one for dad. Each one of these 46 chromosomes they're doubles, though, right? There are 2 pairs of those, and so some will be expressed, some will not, but you'll have a very different look because of this great mixing of the genetic material. It also determines the sex. That's who. You guys know who that is? Slide 4. I guess you can't watch live, but that's. I'll pretend I'm just doing a YouTube lecture for you. That's slide 42. That's George of the Jungle, good cartoon when I was growing up. I think he's still around. Anyway, it also gives you the sex of the little human. The male determines the sex, remember, from your genetics class, which hopefully you've had genetics before you've gotten here. The male determines the sex, because the female can only give, with regard to the 46 chromosomes, one of those, or 23 pairs of chromosomes, one of those is the sex chromosome, and the female is always X. But the male sperm can carry an X, or it can carry a Y, so the male determines the sex. If its sex chromosome is X, you're going to have a female offspring. If the sperm is a Y, its sex chromosome is a Y, you're going to have a male, which is an XY on the sex chromosome. Cleavage. So let's get our little human going, because right now we have a one-celled little human called a zygote, and that's not going to do because we have about 37 trillion cells in a human baby. So we've got a lot of work to do. How are we going to do that work? certainly not by meiosis anymore, but mitosis is going to start happening. Mitosis is still going on. Our cheek cells, our skin cells are still going through mitosis to this very day. But that's how you replicate cells, is through mitosis. And that what happens. And the early process is called cleavage. And we're basically going to split the nucleus of our zygote. We're going to split it in two. And then we're going to split the nucleus into 4s, and then into 8s, and then to 16s. So we're going to split that nucleus over and over and over again. On slide 45 here, just the process of mitosis, which you should be experts with. What the drawings never show you, though, with regard to this process, I drew in blue here. That's the Zona Pellucida. And there's a corona radiata around there as well. So I don't know why that was supposed to be red. I don't know why that turned blue. But anyway, just to remind you, in red here, there's the Zona Pellucida hanging out. And so after the first round of splitting, we actually have a cell with 2 nuclei in it, or two cells within it, which each contain nuclei. So that's what we're doing. We're splitting this ootid into 2s, but the Zona Pellucida doesn't split with it. The Zona Pellucida just stays on the outside. See, we have 2 cells here, but we still have 1 Zona Pellucida. So that's another kind of thing the board's books don't mention, which kind of makes this confusing. So slide 46, meet the 2-celled embryo. So each zygote once the zygote has cleaved into 2 cells, and those are daughter cells, we usually call those, but not in embryology. They're called blastomeres, but they're really just daughter cells. So we're splitting this cell into 2, and those are called blastomeres. But once we've taken the zygote and split it into 2, not replicating the Zona Pellucida, by the way, we get another name change, and that's where the embryo comes. So you could still call it a zygote if you want, but more specifically, once it's 2-celled little human, it's called an embryo. And Morse, which loves throwing weird words out, Morse calls it a conceptus, is a AKA for an embryo. So that's just something you got to stick in your brains. All right, and yep, so what's the genetic count? Each one of these daughter cells, or blastomeres, is a body cell. It's 46 chromosomes, or 23 pairs of chromosomes. They're diploid. Right, let's look at a picture. So here is, you can see the Zona Pellucida has not replicated. Zona Pellucida stays on the outside, but the cells, the zygote has replicated. The zygote has split into 2 pairs, but the Zona Pellucida didn't want anything to do with that. So you can see it has 2 nuclei. Each one of these cells is 46 chromosomes. And yeah, this is the first embryo, so we have a more complex human. Slide 48, sometimes the 2 zygotes split into different ways, or split into itself. So let's talk about twins now, another rabbit hole on slide 48. So there's 2 types of twins, very high yield. There's monozygotic twins and dizygotic twins. Oh, look at that word, zygote. Oh, we know that word, zygote. We just made a zygote not too long ago. So monozygotic twins have 1 zygote. Dizygotic twins have 2 zygotes. How in the world can you have 2 zygotes? Let's look at monozygotic twins first. Well, these are also called identical twins. And I'm on slide, I have the stars blocking the slide view. I'm on slide 50. Monozygotic twins, aka lots of stars here, right? You got to know this. Identical twins. So only 1 sperm was used to create the 2 fetuses. So we have 2 fetuses here. We have 2 zygotes, and we have 2 zygotes and 2 of everything, 2 babies. Only 1 sperm is used, though. So if only one sperm was used, I mean, these twins are going to be identical. The zygote was cleaved, and the zona pellucida. Remember I said the zona pellucida usually is not cleaved? This time the zona pellucida is cleaved into two. And so we have two zona pellucidas. We have two zygotes with their own zona pellucidas. So we have two little humans growing at the same time. And it's thought to occur in the second cell stage here. Okay, and each one of these zygotes then will develop normally, but they have the same genetic material because we split the zygote in half and the zona pellucida in half. Okay, they each implanted the uterine lining, which we'll talk about, and so they develop into their baby. So here's a picture on slide 51. One zygote morphs into a two-celled embryo, right? So here is the first little zygote we created, and for whatever reason, when it splits into two cells, the zona pellucida splits as well. And so that's weird. It doesn't usually happen. But we have two babies. Sue and Sally are born from this process. So each twin will have identical genes. So they'll definitely be the same sex, either females or males. They will look an awful lot alike, maybe not perfectly identical, but very, very similar. They'll have very similar fingerprints, almost impossible to tell apart. They'll have the same blood type. They may or may not develop their own placenta. Sometimes they share a placenta. If they share a placenta, that's called a monochorionic twin. They're monochorionic twins, means they've shared the same placenta, or they could have their own placenta. All right, fraternal twins. My daughter just had this flavor of twins. So two different eggs have been fertilized. So this is not a problem with anything we've been talking about. This is a problem with ovulation. Usually the ovaries take turns. 1 month, the right ovary will ovulate and put out a secondary oocyte. The next month, the left ovary will put out the oocyte. But for whatever reason, sometimes both ovaries put out an egg at the same time. Or maybe one ovary will put out two eggs at the same time. However it works, there's two eggs are going to go through this whole process that we've talked to. And these are called fraternal twins or dizygotic twins, meaning there's two zygotes that have occurred. And so these will be just like normal brother and sister. They'll only have 50% of the same genes. They could be different sex. My new grandbabies, one's female, one's male, and they don't really look alike. And my two babies don't look alike that much. And they have different blood types. I don't know what their blood types are, but they'll have different fingerprints, just like a real brother and sister. So here's the slide 54. Here's the picture of that. And so two separate fertilization events, two separate babies. All right, now another one. This one is incredibly rare. It's called polyspermy. And all those mechanisms, fast block and slow block polyspermy, are to prevent this from happening. You don't want to get two sperms in the egg at once because the fetus won't make it. Almost always the fetus it won't reach that stage. It's too crazy. But polyspermy means two sperm have actually gotten into the egg at the same time. And therefore, we actually have three sets of genetic material in the oocyte, and that ain't good. It creates what's called a triploid zygote. So we have 23 chromosomes from mom, 23 chromosomes from sperm 1, and then another 23 from sperm number 2. So it's incredibly rare. In fact, there's only 2 documented cases that have been allowed to be studied in the medical literature. Of course, there's hundreds more than that haven't been studied, but there's 2 that have allowed study. There's thought to be about 1,000 children that were successfully born that we know about who were actually polyspermy. And again, there's thousands more that slipped by in third world countries and things like that. But there's not many of them. If they do survive, they're called semi-identical twins or susqui-identical twins, which is on slide 56. That's a good AKA, susqui. That's what I would use if I was writing a board question, because you'd have to know that if I say semi-identical, you'll know what it is. But susqui-identical, susqui-zygotic twins, susqui-zygotic twins is the good AKA here. And so, what's the deal with this? Well, all of the body cells will have different mixes of chromosomes. Maybe 33% will be mom, 33% will be sperm 1, 33% will be sperm 2 in some cells. Some cells 80% will be mom, 20% will, or 10% will be sperm 1, 10% will be sperm 2. You can have all sorts of mixes of chromosomes like that. There is the most recent set of these semi-identical twins are from Australia, and we're studying them right now. You can read about them in the literature. But they have different mixes of mom's and dad's DNA all through this different cells that have been allowed to be sampled. And all of the ones that have ever been studied are hermaphrodites. So there's Hermaphrodites, the Greek god of, I don't know what she is, the Greek god of whatever. But she was half she and half he. Has a penis, yet looks like a female, has one breast and doesn't have another breast. So that's because of different mixes. All the body cells have different mixes, so you get the sexual development very, very strange. So hermaphrodites come from semi-identical twins, which is so, so incredibly rare. All right, let's finish up this lecture here. So let's take our little embryo, who's two-stage, and let's get him more toward a human. Let's keep this process of mitosis going. And that's what happens. Each one of these blastomeres goes through mitosis again. So a 2-cell embryo soon becomes a 4-cell embryo. A 4-cell embryo becomes an 8-cell embryo. Just a side note, that the blastomeres aren't always the same size. So they don't always go through mitosis at the same speed. In this case, we have a 9-celled. Here's a picture of a 9-celled embryo. So it's not a perfect, 2 goes to 4, goes to 6, goes to 18, just FYI. When you reach the 8th cell stage, or the 8th cell stage, when there's 8 blastomeres within the Zona Pellucida, something called compaction kicks in, because the cells are getting bigger and bigger up till about the 8th stage, and then they stop getting bigger. They can even start getting smaller and smaller, and that's because these junctional complexes develop in the process called compaction. So you've probably studied these junctional complexes, sometimes called CAMs, cell adhesion molecules. They're already being created. So we have genes now turned on that are making these CAMs. And this picture, you have to know this thing cold for histology, and maybe even me. I've asked this before. There's a star in this one. But tight junction, here's 2 blastomeres side by side. Why are they compacting, getting smaller? Because these have pulled the cells together. We have tight junctions, which we'll talk about in 5th quarter. These are the ones, for example, in the esophagus. These are the ones that prevent acid from getting in between the cells and getting down here where the bloodstream, where the capillaries are. Right? There's the microcirculation lives under these epithelial cells. Tight junctions prevent seepage. They're not very good at keeping the cell together, but we do have the macula adherens, more commonly known as desmosomes, and the zonula adherens. They hold these cells together tight. Gap junctions, that allows for communication, and we got all these other things I'll let Dr. Doe talk about. But yeah, make sure that's what compaction is. That means that these junctional complexes have come to life and are pulling cells in. So our next milestone is arguably the 16 cell stage. When the little human reaches 16 cells, we get a name change, and we call the little zygote or the little embryo, we call it a morula at this point. You can still call it an embryo though. You can even still call it a zygote. But the morula means that point of development where there's 16 blastomeres surrounded by the zona pellucida. The early authors thought it looked like this berry, the small berry. Now, confusingly, some authors call it a morula at the 32 cell stage, which is confusing. Other authors say 16 to 32. So if this is going on YouTube and I have my followers watching this, make sure you look at, if you're a medical student, look at your board, look at Carlson. Chiropractic students, look at Langman or Moore's, see what they say. Unfortunately, Langman says 16 to 32, Moore says 16, so they don't even agree. If I was writing a board question, I would stay away from exactly where. I would say somewhere between 16 and 32. It's probably the best way to say this. But anyway, that's the morula. There's a picture of a morula right there on 64, slide 64. And yeah. What's the slide 65? The genetics, it's same thing. Each blastomere still, even though they're getting smaller, they're still 46 chromosomes or 23 pairs of chromosomes. If we break that 46 chromosomes down, we can say there's 44 autosomes, run-of-the-mill cells, but there's still 2 sex chromosomes, an X and an X, or an X or a Y. And there's the confusion, right? You could say there's 22 pairs of autosomes, or 1 pair, because a pair means 2, right? Confusing, so study that. Make sure you understand that so you're not tricked. Just kind of a big picture of where we've been. So day 1 after fertilization started, after the sperm got through the zona pellucida, day 1 we got 2 daughter cells made, which created the embryo. Day 2, 4 blastomeres are usually present. Day 3, 8 blastomeres. Day 4, 16 blastomeres are usually present. That's called the morula. Alright, and that's enough, definitely enough for your brains are probably exploding with this crazy information. So I will shut this down. This one is going on YouTube, so let me make sure I don't mess this up.